Capuchi-Net, iteration B, network type B, context at meaning and decision stages of cognition, non-platform context, bark alarm call, elicits escape response in quadrupedal motor schema. Non-platform context, hiccup alarm call. Elicits escape response. Capuchi-Net, network B, bark alarm call in platform context. Elicits escape response. Capuchi-Net, iteration B, hiccup alarm call in platform context. Inhibits escape response. Loading network C. Network C, Capuchi-Net, only integrates context at the level of decision and not meaning. Network C, bark alarm call in a non-platform context. Elicits escape response. Capuchi-Net, network C, in non-platform context, hiccup alarm call. Elicits escape response. Capuchi-Net, iteration C, platform context, bark alarm call. Inhibits escape response. Capuchi-Net, iteration C, platform context, hiccup alarm call. Inhibits escape response. You can see the platform there. So to begin with, you've just seen Capuchi-Net in action, and just a little disclaimer, this is what you're expecting to see, but these visual connections from the visual module were removed during the actual simulation, because firstly because a camera was being used, which you could see in that video, and also because the network is very large, as you can see in figure 1, and the network has to remember the thresholds for firing in each neuron. It has to remember the weights that I've adjusted for the connections, and that slows down the processing. So in order to actually run the simulation, I have to actually break down the network slightly. So that's just a bit of a disclaimer there as to why you might have noticed some differences. and of course network B was integrating context at meaning and decision, whereas network C was only integrating context at the decision phase. And I hope you can see that on the video. But so to move into how the network's working, the network dynamics, I have to first talk about how neurons work, so a bit of in computational neuroscience, at least, the theory of how neural networks are enabling cognition to emerge from the brain. And then I'll move into the neuroethology of primates, so how their brain neural network structure and cognition is adapted to producing their behaviours, and why Capuchi-Net mimics some of these cognitive processes with its structure, but doesn't necessarily actually act as a primate brain. Of course, there are many billions of neurons, even in lower primate brains, and Capuchi-Net, depending on which variation, has around about 255 neurons. So it's not really recreating a primate brain, but the function of the primate brain, and I just want to make that clear as well. So any neural network takes as its founding principles that it simulates cognition and the computation that goes on. And what it does is, unlike a computer that will use an algorithm which basically manipulates symbols like it using a mathematical equation, neural networks do computation, but they do it sub-symbolically, which means it's the pattern of activity in the network that represents things, not individual symbols. So just to begin with, the basics are that you have a layer of input neurons, and each layer has a set of neurons, and as you go in a feed-forward classical network, activation will spread through the network from the input to the output, and then that will release the behavior. So in this case here, we've got 3 input neurons. They can either be active or inactive. When they're active, they activate the weighted connection to the next neuron, and as can be seen in this equation here, what the neuron will do is it has a certain threshold, and if the summed input of these neurons multiplied by their weights reaches that threshold, this neuron will fire and activate its weighted function for whatever next neuron it's connecting to. So basically, inside this neuron is this function here, the sum function. What this does is it says add up the activations of each of these X input neurons, multiply them by the weights of the connections, and these weighted connections are important. If you want this neuron to only fire if features A and B are present but not C, you need to make sure that the weight of C is lower, essentially. And then moving on to Rumelhart's classic conception of a neural network, you can have different kinds of sum function. So, different kinds of threshold function. So you can have what's called a sigmoid activation function, which is what's here. It works using exponentials, but basically, the input from several neurons, like this neuron here and this neuron here, multiplied by their weights, could make extraordinarily varied values, and essentially you want this neuron to decide whether it's on or off, like a binary unit. And so a way of converting whatever result you get from that summation process into 0 or 1, you can use a sigmoid squash function, so that exponentially large numbers will still be squished either to 1 or 0. And you can also have other functions. For example, a simple step, binary step function. So that's the basics of how neurons work within the network in simple feed-forward networks. Now on to the neuroethology of primate vision, which is important for Capuchi-Net, obviously. Um, so in 1968, we started, neuroscientists started exploring primate vision. This is the top of a brain, so the superior view, and this is a primate brain. I believe this was either a macaque species or a spider monkey species. And at the back of the brain, the occipital lobe, there's this fold called the lunate sulcus. The brain's got loads of sulci and gyri, particularly in large primate brains. And posterior to this lunate sulcus is the striate cortex. This is involved in vision processing. And these dots are where they put electrodes into the brains of their various species, and they did single neuron recordings to see if what neurons are responding to what visual elements are in the environment. And they also did this in a cat in 1964, and they came across this fundamental computational principle about the vision in mammals. And so I've got a good example. Many aspects of vision are conserved evolutionary amongst species. So I've got an amphibian here. I've got a toad. The neuroethology of vision of the toad is very similar in the early stages of visual processing to primates. So here we've got the eye of the toad, and you've got the retinal cells at the back of the eye. This is the visual field, and it goes through the iris, the center of the iris, and then the light hits the back of the retina. And individual cells here feed forward to bipolar cells and eventually onto retinal ganglion cells. And essentially that's what this schema is representing here. Each of these crosses are single points cells, cone cells of the retina. There's also some rod cells there as well. There's different ratios specifically in the retina, but especially in primates with color vision. But these will then feed forward into those retinal ganglion cells. And so retinal ganglion cells, what these larger circles represent around these X's, is activation for areas of light. So these retinal cells are points of light which together hit the retinal ganglion cells and then represent an on center and then off surround. These larger circles are off surround, and this just basically gives you better visual acuity for understanding points of light. The retina then feeds forward to an area of the thalamus called, in primates at least, the lateral geniculate nucleus, and then from there onto the striate cortex at this region here. But what's Hubel and Wiesel discovered in 1964 with primates, and then with cats, and then again in 1968 with primates, is that firstly that the striate cortex is retinotopic, which means that points of light on the retina correspond with specific regions in the brain, like a map. Retinotopic meaning, retinal map. And that there are simple cells and complex cells, and so basically activations points of retinal ganglion cells, the on center off surround of light in the retinal ganglion cells will be conserved in terms of information processing. Then when they get to the striate cortex of the occipital lobe, they will converge and synapse with the simple bar detector cells of the striate cortex, which means that if you get 3 points of, or in this case 4 points of light in a row, they will all sum their activity and cause this neuron to fire because it meets its threshold. And so this neuron is now firing only when there's an entire bar of light. So this neuron detects bars of light, which is shown by this dotted line going around here. And so simple neurons in the striate cortex detect bars of light, essentially. and then these then feed forward onto complex cells and then hypercomplex cells, which sums bars of light together into more complex shapes. And these were the principles that I used when making Capuchi-Net on Neuronify 1.2. It's a great piece of software if you just want to basically simulate a neural network without using statistical methods to try and train it, using backpropagation, more complex computational algorithms as well. It's an educational tool. Ideally, you'd want to move on to something on MATLAB in the future. So essentially, you've got retinal ganglion cells, and these will then three of these synapse onto each of these. So each of these, what you could imagine as striate neurons, simple neurons in the striate cortex, these are responding to bars of these three retinal ganglion cells active, and they only respond to those. And then those feed forward onto more complex cells. And what these neurons here, these artificial neurons, will only fire in the presence of things like wings, talons, heads, bodies, legs, things like that, tails. And then these will then feed forward through the network, getting more and more complex until eventually you get the representation of entire animals in the visual field. And that's important for Capuchi-Net in order to represent at higher areas of the network predators and conspecifics, as well as the platform. And I will demonstrate how the neural network actually has the ability to process these things. And that's why we can, at higher levels of the cortex, just say that individual neurons are representing raptor and felid, snake, and capuchin. In terms of the auditory system, sorry, like I said, the reason why I removed this during the simulation is it slows down the computer something chronic. There we go, we've scrolled up now. I essentially mimicked the layout of the cochlea and the primary auditory cortex. Because the cochlea constricts in a spiral, it means that when you hear a sound wave and its component frequencies, the cochlea actually splits up the component frequencies naturally just by the way of its architecture, so that you get low frequencies earlier on in the cochlea and then higher frequencies as it spirals in, which means that neurons with hair projections processes can actually represent individual sound frequencies, and these then feed forwards. Instead of retinotopy, we talk about tonotopy. So individual tones represent different spaces in the primary auditory cortex. And I can show you an image of that here. So this is the temporal, superior temporal lobe of a primate brain, and you can see here that as you ascend upwards along the auditory cortex, you get higher frequencies. So this essentially enabled Capuchi-Net to classify a bark and a hiccup differently according to the structural principles of their component frequencies. So you've got this neuron would represent 0.1 kilohertz, 1 kilohertz, 2 kilohertz, 4 kilohertz, 8 kilohertz, 1632. And I'm just going to stop now, and then I will show you how Capuchi-Net can identify objects in the visual field. To go from predator representation to escape response is probably going to take a while, so you might have to be a bit patient there. You can see how long it's taking just to open the file, and on top of that my computer's having to deal with doing a screen recording. Yeah, you can probably hear the fan on the in the microphone going round right now, so I'll make it brief. So this is the part of the network we dealt with cognitive pragmatics. So you've got the cochlea, you've got the bark and the hiccup alarm calls. You have going from the top raptor, and then you've got felid, and then you've got snake, and then you've got this neuron here, which is an inhibitory neuron. Well, it's a neuron that has an inhibitory axon synapse, which is the platform that activates the feeding platform. And then lower down here you've got capuchin, and then you've got the posterior cingulate cortex connection to the prefrontal cortex, which is, you know, when the primate gets worried because it's seen a predator, it makes a decision to elicit an escape response, which is what these 4 neurons are here. They have afferent connections to muscles, hooked up to produce quadrupedal locomotion like pattern. And then you've got the visual system, which we addressed in a previous video as well. So without further ado, we're going to press Raptor, which should make a raptor icon appear on here. The previous video to this shows you what the raptor icon should be. And then the neuron that we should see firing is the one up here for raptor. And then this should activate this and elicit an escape response up here. So let's hope that the processing of my computer is up to the challenge. Fingers crossed. Here we go. It's taken a while to even register that I've pressed anything. Oh, there we go. Raptor. Come on, we can do this. Come on, Capuchi-Net. Literally, your life depends on running away from this raptor. Okay, I'm seeing some neurons in layer 1 firing, I think. There we go. Some just fired there. It's difficult to see from this far away, zoomed out from the network, but you have to be. Otherwise, you can't see what's going on in the entire network with it being so massive. And there, layer 2's just fired off. The next layer is something akin to what you'd find in the parietal lobe, which is where in the visual field it's more to do with motion tracking and spatial analysis. Can't see a so one just fired off at the bottom. So now look at that raptor neuron. The one that's over here. Watch out for this guy firing off. Yes, raptor neuron just fired, so it's recognized as a raptor there. There should be a decision about it being a negative thing in the environment. There we go. So that's something that's like biosemiotics, where, you know, the animal is immersed in a semiosphere and it has to interpret signals within its kind of niche about whether the signal is good or bad. There we go, and we've got that's initiated the escape response. In the last video I did, we focused on predator escape responses, but I totally neglected how Capuchi-Net deals with conspecifics, i.e. other capuchins in the social group. And that's equally as important, because depending upon the kind of interaction you're having with another member of the social group, you may want to run away to avoid displacement or agonism from a higher ranking individual, or you might want to give grooming to another individual. But at the fundamental basis of this is recognizing a conspecific, and dealing with a conspecific different to how you deal with a predator, for example. So we've got Capuchi-Net loaded in front of us again, and just a reminder that this neuron here is going to be representing raptor, felid, snake, feeding platform, and then below it is capuchin, or in this case it will be conspecific, and the predator neurons excite the decision module into producing an escape response. However, if we're dealing with a conspecific, like a high-ranking individual, you might want to groom them instead. And so Capuchi-Net shouldn't produce an escape response to a conspecific. Alternatively, it could be a mate, in which case you wouldn't want to be running away from it, you'd be trying to attract it. So we're going to be pressing the capuchin light signal, which should activate the representation for capuchin. You can look at visual object recognition, the video I did for that to learn what the icon is, but you should be able to see it on here. And then the signal should propagate through the network, and we should see this lower neuron here activate, and there to be no escape response up with the motor neurons over here. So that was capuchin activated on the retinal ganglion input. We're looking at layer 1 now. So that's now activated, and that computes linear angles, horizontal, vertical, diagonal right, diagonal left. And we're looking here. These neurons are firing. They identify corners and features such as legs of quadrupedal animals like the legs of the capuchin, the head and the tail. Now we're looking for this neuron to fire over here, where I've got my cursor, the capuchin neuron. And it's just fired. And no escape response. So that's the equivalent of Capuchi-Net recognizing that there is a conspecific in the environment.